Hello students, today we are going to learn about conjugation as gene transfer method. As such, there are transformation, transduction and conjugation are the three commonly used gene transfer methods in bacteria. And today we are going to learn in details about conjugation as bacterial gene transfer method. So the bacterial conjugation or mating is a mechanism of genetic transfer that involves cell-to-cell contact. And this mechanism is encoded by a plasmid, which is known as a conjugation plasmid or a conjugative plasmid. In general, there are two types of cells involved in the conjugation process. One of the cell is known as the donor cell, also known as the F+ cell, which contains the conjugative plasmid. And the other cell is the recipient cell, which does not contain the plasmid and is also known as the F- cell. F here stands for fertility. The transfer mechanism may differ depending upon the plasmid involved, but in most gram-negative bacteria, similar mechanism as that of transfer of F plasmid is being employed. So here we can have a look at the map of a typical F plasmid. The size of the plasmid is about 92.2 kilobase pairs. It has origin of vegetative replication, which is used for normal replication within the cell, by which the number of plasmids per cell can increase. Then it has an ori T, which is known as origin of transfer. So this is the region from where the transfer of genes will begin when the conjugation process takes place from the donor cell to the recipient cell. Then we have a TRA region, which contains genes that are required for the transfer function. And these involves genes for synthesis of cell surface structure that is known as a conjugation tube or the sex pilus. Apart from this, there are presence of these insertional sequences that you can see in yellow. I represent as IS2 and IS3. So these are the sequences or regions which are complementary in sequence with the bacterial chromosome, and they can actually integrate into the bacterial chromosome by recombination between the insertional sequences and similar sequences present on the chromosomal DNA, resulting in formation of something which is known as an HFR strain, which stands for high frequency of recombinant strain, which basically consists of a strain in which the F plasmid is integrated into the genome of the bacterium. So this next slide talks about the mechanism of DNA transfer during the conjugation. So the process is initiated by the F+ cells, which will extend its pilus, and then it will attach to the F- cell. Retraction of pilus then takes place so that the two cells, the F+ and the F-, are present in close proximity to each other. Once their interaction is stabilized by certain cell surface proteins, which are also associated in the conjugation process, the process of conjugation will begin. Now, during the gene transfer process, first there is formation of nick or a break within one of the strands of DNA, that is the outer strand of DNA, while the inner strand or the inner circle of DNA of the plasmid will remain intact. Now, from the nick region, so this nick will happen at the origin of transfer region, where we had seen the arrow mark in the previous slide. So the DNA will start entering into the recipient cell, beginning at the ori T sequence. As the outer strand of plasmid DNA is entering into the recipient cell, the bacterial replication machinery will start functioning, and it will simultaneously extend the outer strand of the plasmid DNA such that one length of the plasmid DNA is stripped out as it is entering into the recipient cell, and simultaneously one length of the plasmid DNA is synthesized such that at the end of the process, double-stranded plasmid DNA will be present in the donor cell. Simultaneously, as the single-stranded plasmid DNA is entering into the recipient cell, the recipient cellular machinery will also begin synthesis of the second strand of the plasmid DNA. Thus, at the end of the process, both the cells are going to have a complete plasmid. And in this way, now the recipient cell, which was originally F minus, will now be getting converted into an F plus cell. So this cell is capable of mating with other F minus cells. Since it has the complete conjugation plasmid, it can transfer the plasmid to other F minus recipient cells. The next slide talks about formation of HFR strains and how it leads to mobilization of chromosomal DNA. So, this in purple is the F plasmid, and within the F plasmid are present the insertional sequences. These are the regions of homology between the F plasmid and the chromosomal DNA. So, the one in green is the chromosomal DNA. So, once the plasmid DNA has entered inside the recipient cell, it can stay as a plasmid itself, or it can also integrate into the chromosome. So if it integrates into the chromosome, this integrated form of the F plasmid is known as, that leads to generation of a strain which is known as an HFR strain, that stands for high frequency of recombination strain. So here, all the genes that are required for the conjugation process are present within the cell, the HFR cell. However, the plasmid is actually integrated inside the genome. Okay, so now when the conjugation process will begin, one of the strand, that is the outer strand, will form a nick at the origin of transfer sequence, and it will start entering, as if you can see in this diagram, it will start entering from here into the other F minus recipient cell. Now, as a result, as the conjugation process will continue, after transferring half of the plasmid DNA, the next sequence that is present adjacent to it is a chromosomal DNA. So after that, it will start transferring the chromosomal DNA. However, the chromosome is quite huge compared to the plasmid, and the process of conjugation is not very long, and the conjugation tube or the connection between the two cells often breaks after a few minutes of conjugation. So when the connection breaks, whatever part of the chromosomal DNA has been transferred after half of the plasmid DNA, that will enter into the recipient cell. And because of homology, again, the transferred DNA can recombine with the recipient cellular DNA, and that will lead to generation of a new phenotype. However, the recipient is never going to receive the entire plasmid whenever there is a conjugation with an HFR strain. This is because the process is not long enough to completely transfer the half of the plasmid and the entire chromosome, and then the remaining half of the plasmid. So as a result, the recipient cell is not going to become again an F plus strain, or it will not become an HFR strain which can further transfer the genes to other recipient cells. Only it will have high rate of recombination because it is receiving it is receiving the genes from the HFR strain, the chromosomal genes it is receiving at a high rate, and hence there will be a lot of recombination reaction, and that will lead to new phenotype. So that's it about conjugation. Kindly like, share, and subscribe if you like the video, and as well as to get the direct update whenever I upload a new video. Thank you.